chemicals naturally flow down a concentration gradient, moving from high to low areas. To stay alive, a cell must frequently reverse this flow, which pushes matter back against an immense chemical current. Moving substances against their natural gradient requires physical work. It necessitates a highly specialized, energy-intensive mechanism to drive the effort. Primary active transport is defined by how it powers this work. It is a transport mechanism where energy is liberated directly from the breakdown of ATP. Adenosine triphosphate, or ATP, provides the chemical energy that powers this specific process. This method is used to move vital substances across the cell membrane, including sodium, potassium, calcium, hydrogen, and chloride ions. Without a system to directly couple chemical energy to physical movement, cells would reach equilibrium with their surroundings and be unable to maintain the internal chemical gradients necessary for life. Specialized carrier proteins embedded in the cell membrane act as microscopic engines, performing the physical work of transport. This diagram shows three prominent examples of these engines: the sodium-potassium pump, the calcium pump, and the proton or hydrogen pump. The sodium-potassium pump, or Na+, K+ ATPase, serves as the most critical model for understanding how this class of protein operates. To see how chemical energy translates into mechanical work, we must analyze the physical architecture of this specific pump. Examining the structural configuration of the sodium-potassium pump reveals the universal rules governing how a cell manipulates its internal environment. The carrier protein of the sodium-potassium pump consists of two distinct protein subunits, one alpha subunit and one beta subunit. This model isolates the smaller component embedded in the membrane, the beta subunit. It is a glycoprotein, and its role in transport is secondary to the pump's primary work. The larger alpha subunit acts as the primary workhorse of the mechanism. There is a strict division of labor here. The actual physical transport of sodium and potassium ions occurs only through the alpha subunit. In cellular machinery, form strictly dictates function. The size and complexity of the alpha subunit are strictly required to manage the physical transit of charged ions. The alpha subunit relies on a highly specific internal geometry containing distinct receptor sites to identify its targets. On the inner surface facing the cytosol, the protein possesses exactly 3 receptor sites shaped specifically for sodium ions. On the outer surface facing the extracellular fluid, there are exactly 2 receptor sites designed for potassium ions. This precise stoichiometry is hard-coded into the protein. It is built to consistently handle 3 sodium ions from the inside and 2 potassium ions from the outside. This source confirms the location of the outer potassium sites and introduces the final structural component, a site for the enzyme ATPase. The ATPase site is positioned near the inner sodium binding sites. The protein functions as a mechanical gate, requiring the simultaneous binding of 5 specific ions, 3 sodium and 2 potassium, before the transport cycle can progress. The transport cycle remains paused until all 5 receptor sites are occupied by their respective ions. Reaching this state of full occupancy is the trigger that activates the nearby ATPase enzyme. Once activated, the ATPase enzyme captures a passing ATP molecule. The enzyme splits the ATP molecule into ADP, which drifts away, and a high-energy phosphate that firmly attaches to the pump. This localized energy release causes the alpha subunit to undergo a massive structural shift, known as a conformational change. The structure contorts, closing the inner pockets, opening to the opposite side, forcefully ejecting the 3 sodium ions and pulling the 2 potassium ions inside. Through this precise interaction, the chemical energy stored in the phosphate bond is converted directly into mechanical movement. This sequence, binding, ATP breakdown, structural shift, and release, is the mechanical stroke that allows the cell to fight the force of diffusion. Is this primary active transport mechanism restricted only to the outer cell membrane? Evidence from calcium transport suggests otherwise. This process is operated by a separate, specialized carrier protein, but still relies on ATPase to function. While the hardware of the calcium pump is different, it uses the exact same principles of ATP hydrolysis to perform work. These calcium pumps operate deep inside the cell, specifically moving ions into internal organelles like the mitochondria and the web-like sarcoplasmic reticulum. In muscle cells, these pumps actively sequester calcium ions inside the sarcoplasmic reticulum. Nature uses the same primary active transport blueprint, direct ATP energy coupling, but swaps out the carrier protein hardware to manage different ions in different parts of the cell. Primary active transport is defined by its mechanical requirement, the direct localized liberation of energy from ATP via an ATPase enzyme. Executing that exchange depends on precise protein architecture, such as the rigid 3:2 stoichiometry of the sodium-potassium pump. To maintain a functional baseline, millions of these pumps fire relentlessly every second, executing perfect mechanical strokes across vast microscopic landscapes. While the sodium-potassium pump manages the cell's global boundary, specialized units like the calcium pump manage highly localized internal environments. This diversity of pumps provides the cell with targeted control over its chemical environment. Without this constant expenditure of chemical energy, internal gradients would collapse and cellular function would cease. Primary active transport establishes the electrochemical conditions that allow every other cellular process to function.